Hello, everybody. My name is Brian Kepler. I'm director within our Discovery and Translational Sciences team within Metabolon. I've been with Metabolon for about 12 and a half years within our Discovery and Translational Sciences team. Today, I'm going to walk you through what our typical deliverables are for our global untargeted biochemical profiling. There are typically two deliverable files that are going to contain your data. The first one is the global data table, right? The global data table is going to be an Excel file. It's going to be separated into multiple tabs. The first tab, as you can see here, is simply a data key and explanation tab, which will describe what you're going to be looking at and the tabs to come. The chemical annotation tab is going to be a list of all the biochemicals detected in your study, as well as a lot of information about those biochemicals. It's going to give information such as the chem ID, the name of the biochemical, as well as a number of different links to things like the HMDB database, the KEGG database. And when I get to that tab, I'll go over some of those specifics. The second tab is going to be the sample metadata, which is basically client-provided metadata for each sample. And this can be very simplified. It can simply be a subject ID and a group number, or it can contain a lot of metadata that you want correlated to your metabolomics data. So that's really directed by the client and is just simply uploaded from the client manifest. And then getting into the data itself, the data is going to be spread across a number of different tabs. The first tab being the peak area data. The peak area data is basically the integrated area under the curve, the total ion counts, if you will. It's the unnormalized peak areas. And these are going to range, depending on the abundance of a metabolite, from, let's say, 10,000 ion counts to 10 million ion counts. So that's what we refer to as basically the raw data. Now, that raw data is going to be adjusted in certain ways for visualization purposes, to look at plots, and also for statistical purposes. Now, we also perform batch normalization of the data. And the reason that we do batch normalization is because there might be variability for different metabolites based on the number of samples you have, based on day-to-day variability, batch-to-batch variability. And this is going to be dependent on the number of samples in your study. So batch normalized data is basically on a per metabolite basis. The raw values from that peak area data are going to be divided by the median. And that's, again, on a per metabolite basis. The median of those samples in each instrument batch is going to be given a value of 1. And then every sample for each, again, on a per metabolite basis, is going to be adjusted based on that. There's also some additional information on batch and batch correction on this information tab below. The next step is to impute the data. This is done to basically remove missingness. And there's oftentimes missingness in metabolomic data. Missingness can be for a variety of reasons. It can be below the limit of detection. This can be due to the amount of sample analyzed, the specific compound abundance, compound reactivity. But as you'll see, as I move into that tab, you'll see certain cells for certain samples on a per metabolite basis are going to be missing. Now, the trick is, how do we deal with that missingness? It could be, really, there's 3 types of missingness. It can be missing at random, missing not at random, missing completely at random. And in order to move forward with the data and perform stats, there are a number of different methods, imputation methods, that can be performed. And Metabolon has done a lot of research on this, on using the minimum, the half minimum. Different people might use k-nearest neighbors, singular value decomposition. There's a number of different areas. And there's also methods to evaluate the imputation accuracy, such as normalized root mean squared error. Now, the method that Metabolon has decided to use is imputing with the minimum, as I said, and we have publications that we can share with you on our decision for that and the justification for that. So after the batch normalized imputed data is performed, there is a possible step, depending on your matrix that you're using, whether we use a normalization to something like protein or DNA, osmo, let's say if it's urine, mass if it's a tissue. So that would be the next step, normalization to some sort of metric like that. And then the last step for the data is the log transform data. Now, Metabolomics data typically displays a log normal distribution. So the log transform data is what is used for statistical purposes. Now this data key and explanation tab goes into a number of different additional explanations on what the different fields you'll see in the tabs to come. I won't go over each individual one of these here on this tab, but I will touch on them as I go through the individual tabs. So as I said, the first tab here is the chemical annotation tab. This tab is going to be, again, a list of all biochemicals identified in your study. So the list of biochemical names is going to be shown here. We also simplify that. If it's a very long name, sometimes those are simplified to a plot name, the names you would see on the different plots. The most important probably piece of information beyond the biochemical name on this tab is the Chem ID. The Chem ID is basically a unique identifier for each biochemical. These are going to be listed on the top row in the coming tabs so that you can attribute the different Chem IDs for each sample to the different biochemical names. Chem ID is also a universal identifier that is used in the field of metabolomics, so you can compare the data from your Metabolon study to other studies using those Chem ID values. Beyond the biochemical name, we provide the super pathway that biochemical is part of, the sub pathway as well. We also provide things like Inchi and SMILES, which would give you an idea of the structure of that biochemical. Some species of biochemicals, such as lipids, are going to be able to be further defined by their species level, their molecular species level, or their SN positional level, as you can see in this one here. We also provide a number of different, again, universal codes that are used in the metabolomics field, CAS numbers, ChemSpider. As I mentioned, links to the Human Metabolome database and the PEG database, the Kyoto Encyclopedia of Genomics and Genome, as well as PubChem IDs. This final column here is the platform or the method of Metabolon's global platform for which that metabolite was identified on. So again, this tab is not going to contain any data. This is simply the chemical annotation, some information on all the biochemicals identified in your study. The next tab, sample metadata. Again, this is not going to contain any data itself. This is just simply a manifest of samples sent to us by our client. This is going to be a lot of information taken directly from the manifest that we receive from our client. So things like the client identifier is going to be taken, the group that maybe is attributed to that sample, usually for statistical purposes, a sample number, subject ID, all that would be provided by a client. Now, Metabolon is going to attribute a specific parent sample name ID. These are simple demo numbers here. An alphanumerical number will be given to your client ID, and it is this parent sample name, which will be used on the subsequent data table, the data tabs that we'll go into next. As I mentioned, this can be as small or as big of a tab as you want. If you want to send things like gender, BMI, any sort of metric you want to use for correlation purposes, you can include that in your manifest, and we would include that here. So now moving to the data itself. The first tab, the peak area data, as I mentioned, this is the total ion counts. This is basically the area under the curve. Now, this is organized in such a way so you're not looking at too much information at once. And this is a number of years ago, we moved to this format of data for a number of reasons. Some client feedback and also our own internal research allows this format to move forward with the data in a much easier way. If you're going to use R for statistics, if you're going to use Python, if you're going to build larger data lakes, this format is probably the best in order to do that. It trims out all the things like biochemical names, all the metadata, and just looks at sample number and Chem IDs. So again, looking at parent sample name, this is all the samples using Metabolon specific identifiers. And then on the top row across here, these numbers, these are the Chem IDs, right? So, and then here would be all the peak area data. As I mentioned before, these are going to be very sample specific, but also metabolite specific levels. Moving forward, as I mentioned, we provide a number of different tabs to better use this data for visualization and statistical purposes. The first being batch normalized data. As I mentioned before, the batch normalization is to really remove batch-to-batch variability and set the median sample value on a per metabolite basis to 1. So as you can see, if we look at some of these, you can see they're no longer in tens of thousands or hundreds of thousands or millions of ion counts. These are all right around 1 or just above 1 or below 1. And it is actually this batch normalized data that is used for our plots. So rather than having plots with many different scales on the y-axis, it's going to be a very similar scale for different metabolites. Now, as I mentioned, you're going to see some missingness in metabolomic data. That's not uncommon. It's actually pretty typical, pretty common. You can see a number of these samples for this specific metabolite with chem ID 932 are missing. As I mentioned, this is going to be for a variety of reasons. It's simply below the limit of detection. It could be sample type dependent, sample amount dependent. If we only received a minimal amount of biofluid or tissue amount, you're going to see a drop in the detection rates of certain metabolites. So, and as I mentioned, the missingness and imputation method we use is going to be imputing with the minimum. So shifting from this batch normalized data tab to the next tab, which is the batch normalized imputed tab, you can see I'll just scroll back and forth here. This row here you see is now imputed with the minimum value for that specific metabolite. As I mentioned, there might be an additional tab in here if you're going to normalize by protein for cell pellet analysis, if you're going to normalize by volume, if you're going to normalize by weight, if it's a tissue, there might be an additional tab in here. But then the final tab is going to be converting this batch normalized imputed data to the log transform data. As I mentioned, for statistical purposes, due to the nature of metabolomics data, it is the log transform data that is going to be used moving forward for statistical purposes. So as a whole, anybody doing a metabolomics study on our global platform will receive a data table exactly like this. If you also contract with us to do statistics, you will receive an additional file, which is going to be our heat map. Moving to that heat map file, it's going to start off with a very similar tab, that data key and explanation tab, because a lot of that same information from the data table is going to be on this heat map tab on the next tab. Simply for convenience, for client convenience. So if we take a look at the data heat map tab, this is going to be obviously a statistically driven tab listing your metabolites, the heat map driven color coding, and that color coding explanation is actually going to be here on that explanation tab. Anything green is going to be decreased, anything red is increased. We also have 2 different shades of green, 2 different shades of red. Typical p-value cutoff is 0.05, so anything less than or equal to 0.05 is going to be that dark green or dark red if it's decreased or increased. We also identify what we call trending changes, so not quite a robust statistical change to reach that 0.05, but it is between 0.05 or 0.1. Those are going to be shown in the light green or pink colors. We also have color coding for ANOVA main effects. I'll touch on that on the next tab as well. Significant p-value of 0.05 is dark blue, light blue would be between 0.05 and 0.1. So moving back to the heat map tab, all the biochemicals are going to be listed here, not alphabetically, but by pathway. The reason we do this is because it's much more easily visualization wise to see changes on a pathway basis when you organize them by pathway than if they're, let's say, alphabetical. So here you'll see the biochemical name, the subpathway, the superpathway for which it's associated. You'll see the platform or the method on our global platform for which that specific metabolite was identified. You'll see that compound ID, which will correlate with the compound ID that you saw on the data table. We also include these same KEGG links, HMDB links, PubChem IDs as well, if you quickly want to get some information on what those are. And then you have your statistical comparisons themselves. The statistical comparisons are really going to be dependent on the study design. Our DTS team will consult really from the start of a study idea to understand what your objective is, help you design the study, attribute groups, and then we can have a discussion on the most appropriate statistics. We can do any number of statistical techniques. If it's simple 2 disparate groups compared to each other, it might be a simple Welch's t-test. If you have longitudinal sampling of the same animal, the same human, it might be something like match pairs t-tests or repeated measures ANOVA, etc. This is going to be looking at repeated measures ANOVA contrasts. So you have your contrast here. You also have the main effects here. And again, as I described and is explained on the data key and explanation tab, you have the color coding. Again, dark red, dark green are going to be significant. The pink or the light green are going to be those trending changes. And the same for dark blue or light blue on for the ANOVA contrasts. So this is going to be the data that most of our clients are interested in, and how significant of a biological change are we seeing in our different groups. However, there is a lot of additional information on this tab, this heat map tab, that can be very useful and informative as well. Obviously, we provide the p-values and q-values for all of these different comparisons. We also provide the mean values that are used for this. And then at the end here, simply breaking out the superpathway, subpathway, ChemID, CAS-ID as well. But this is a pretty significant piece of information that I wanted to explain here, the percent filled values. So these percent filled values, you can see it's broken out for each group, each statistical group. This is the percentage of samples in each group for which each metabolite was identified. So you can see here for glycine, 100% of samples in each group, glycine was identified, right? If you move down to this metabolite here, you can see only 15% of the samples in group Met A actually had this metabolite, so on and so forth. So this is a very informative piece of information to know. Let's say if you see a metabolite that's 100% in a disease population, but 0% in a healthy population, knowing that can be very informative if you're looking for a biomarker, for example. So that is an overall look at the heat map. As I said, if you're any client doing data on our global platform will receive the data table. Anyone doing stats will also receive this heat map. That will conclude what I wanted to go over on this webinar. And as always, if any of our clients want some further discussions with any of our salespeople or anyone within our DTS group, we'd be happy to do that. And we look forward to working with you. Thank you.